In this video, we'll go through the process of discussing how proteins become denatured. When a protein becomes denatured, that simply means that some of the interactions that were present in the tertiary level, so those interactions between the side chains, they've now become disrupted. So let's say we had a protein and we were looking at an interaction between, we'll just say serine and threonine. So instead of drawing out the entire amino acid, I'm going to simply focus on the side chain, because we're looking at what happens to the interactions that were present in that side chain. So if we have serine, we have threonine, they're both polar, so they're both hydrophilic. And if we look, even though we don't see the non-bonding electrons, each oxygen has a total of 4 non-bonding electrons. So when we look at the interaction that occurs between serine and threonine, because they're both polar and they both have hydrogen bonded to oxygen, they both have non-bonding electrons on the oxygen, they can both form hydrogen bonds with one another. So this is the interaction that we will see in the tertiary level. They will form a hydrogen bond. But let's say you decided to add, let's say, a solution of, I'm going to say an aqueous solution of sodium chloride. So of course sodium chloride is an ionic compound, so salt water. But when that sodium chloride is in an aqueous solution, it doesn't actually exist as a compound. Instead, it exists as an ion. So now you have these ions in the presence of these polar molecules that are present in this protein. So if you think back to the intermolecular forces that you learned about in 1151, you learned about London dispersion forces, you learned about dipole-dipole forces, you learned about hydrogen bonding forces, but you also learned about ion dipole forces. London dispersion, those were nonpolar. Even though they are present in every covalent compound, nonpolar that's the only interaction they have. Dipole-dipole occur between two polar substances. Hydrogen bonding occur between polar substances, but specific types. So just put that asterisk there. But ion dipole occurred between an ion and something that was polar. This is in the order of increasing strength. The ion dipole is the strongest. So we have hydrogen bonding, which is pretty strong. It's the strongest of covalent. But when we add something that has ions present, you can now have competition. And so when those ions are present and they're floating around in that solution, they can compete with the serine or the threonine. Serine and threonine, when they're by themselves, they're going to form that strong hydrogen bond. However, with the addition of these ions, you now have competition, because you can form an even stronger attraction between this polar substance and this ion. You can then form that ion dipole attraction. So because of the competition that has now been introduced to this protein, this causes the protein to become denatured. So in other words, it causes different interactions to form. So instead of the serine and the threonine being attracted, that threonine may now be attracted to that ion. Same thing with the serine. It may now be attracted to that ion. And so when this happens, because these two amino acids are no longer attracted to each other, but instead they're attracted to other things, it causes the shape of that protein to change. Because remember, in the tertiary level, the protein folds in order to form those interactions between the side chains. But those side chains no longer have interactions with one another, but are now attracted to other things. The shape is now modified. When the shape changes, the protein no longer behaves as it once did. So therefore that protein becomes denatured. So adding in ions. So whether that ion comes in the form of simply an ionic compound, which we can call that a salt. So table salt is not the only salt. Salts are just ionic compounds. But remember, acids and bases also form ions. Regardless of whether it's a weak acid, weak base, strong base, they still form ions. So the addition of any of these substances introduces ions, which can then cause competition with the compounds or the molecules that are already there. So this can cause the protein to become denatured. Because you have now reduced the number of hydrogen bonds that were present, so you've disturbed those hydrogen bonds, think about which levels contain hydrogen bonds. So not only would you see a competition for hydrogen bonds in the tertiary level, but remember hydrogen bonds were also in the secondary level. So some of those may have also been disrupted. If the protein has a quaternary level, it will also be disrupted. The only thing that does not change by adding that ionic compound, the acid or the base, is the primary level. The primary level of protein structure will never change by simple denaturation. The tertiary level will always change. The secondary level may or may not change. The only time the secondary level changes is if it's a hydrogen bond that was affected. Because remember, in the secondary level, only hydrogen bonds are present. So if hydrogen bonds are now being disrupted, then the secondary level changes. If no hydrogen bond is disrupted, the secondary level will not change. So let's look at another one. So let's say we were now looking at the interaction between, let me just say, alanine and another alanine. So alanine simply has a CH3. Both of these are nonpolar. So if we look at the interaction between them, we simply have that nonpolar or hydrophobic, whichever one you want to call it. They basically mean the same thing. Interaction. If I decide to add an acid to this, it's not going to affect a nonpolar interaction because those ions from the acid are not trying to interact with the nonpolar substance. They're not attracted to the nonpolar substance. Those ions are only attracted to other ions or things that are polar. They're not attracted to nonpolar. So the addition of an acid, so if I decide to add an acid, no change. Not for this specific interaction. Let's say I decided to increase the heat. So I'm going to say extreme heat. Remember, heat is a form of energy. These nonpolar interactions are basically the London dispersion. They're the weakest. They don't require as much energy as the others to be disrupted. So if you add extreme heat, that heat gives enough energy to break certain intermolecular forces. It would definitely disturb your London or London dispersion forces. However, depending on the heat, and since I said extreme, that is also going to be enough heat to disrupt hydrogen bonds. So we would see a change. So a change would occur in extreme heat because this is a weak interaction. So that energy is going to be able to break that weak interaction. Because we are seeing a change in this interaction, we could think about what levels will be disrupted. So I'm going to just put up all four. 1, 2, 3, 4. So primary, secondary, tertiary, quaternary. Again, we're denaturing. No change to the primary. The only way you can change the primary is by hydrolysis or condensation. So a chemical reaction must take place to change the actual primary. Denaturation is not a chemical reaction. Secondary level for this would not change. The reason why? No hydrogen bonds are being disrupted. Tertiary level will always change. So the tertiary level is where you see all these different interactions. So because we're disrupting hydrophobic interactions, which are located in the tertiary level, they would change. If this protein had a quaternary level, it would also change. Because again, the quaternary accounts for the primary, secondary, and tertiary as well. And so because the tertiary of one changed, the tertiary of the other also changes. And so when you're talking about denaturing proteins, simply ask yourself, what does it mean for this substance to be added? What does it mean for this change to take place? Can that change result in competition between what's already there? So again, if you see the acid, or you see the base, or you see the ion, remember that when they're in an aqueous environment, they have ions. Can that ion cause competition? If so, the protein will be denatured. If not, as we saw here, the protein will not be denatured. And so denaturation is basically a competition in order to form stronger intermolecular forces, or even similar, the same intermolecular forces. So if you decided to add, for instance, soap to this, or detergent, soap is polar and nonpolar, primarily nonpolar. So adding soap would cause competition between the nonpolar interactions that were already there. So you're simply looking at whether or not competition can take place. If so, the protein will become denatured. If not, it will not become denatured. If it becomes denatured, ask yourself, does that occur in the primary level? Does it occur in the secondary, the tertiary, the quaternary? If it does, that level will be disrupted. So it'll be altered or changed. If it does not, it will not. For denaturation, primary will never change. Secondary will only change if hydrogen bonds are disrupted. Tertiary will always change. If the protein has a quaternary structure, it will always change as well.